Hi, hello everyone. Welcome back to my channel. Myself Varshini. I'm currently pursuing my engineering at Sri Venkateswara College of Engineering, Bangalore. My USN is 1VE23CY052. My course is CSE for Cyber Security. Semester is 4th semester. Let us study in this video is a biology for engineers. Let's go for next slide. Introduction to DNA vaccines and diagnosis. A DNA plays a critical role in the modern development of our vaccines. A significant for a diagnosis method for various diseases. The understanding DNA function enhances our ability to combat infections and diagnose conditions effectively. A DNA utilizes a genetically engineered DNA to provoke an immune response. They are designed for and instruct cells for producing antigens that are immune part of pathogens. This technique is a promising platform of a rapid vaccines of developing against the emergency diseases. Let's go for next slide. Mechanisms of the action. DNA vaccine contains a genetically engineered DNA that codes for the antigens. These antigens provoke the immune response without a presence of actual pathogens. This innovation approach has shown a promising against the various infection designs and cancers. The DNA has a vaccine entire host cells and it is translated into a messenger of RNA, mRNAs also. Then mRNA is then translated into proteins which are recognized as a foreign by the immune system. This process simulated both cellular and human immunity, providing a proteins against the infections. This is a mechanism on actions of the DNA. Let's go for next slide. Example of DNA vaccines. The YCOD vaccines used for a COVID-19 is an example for DNA vaccine. The other DNA vaccines are developing for the diseases like Zika and West Nile virus. The clinical trials are going on evaluating this efficiency and safety across the various populations. Let's go for next slide. RNA vaccines for COVID-19. It represents a background-breaking approach for a vaccination that have been at a forefront for effort of a combining of pandemic. These vaccines are utilized for messages of RNA, mRNA to instruct as cells in a body to produce a protein like as spring proteins found in on surface of a SARS-CoV-2 virus which causes COVID-19. The immune system that is recognized to spring a protein that mounts a response including production of antibodies and to protect against future infections. The RNA vaccines for the COVID-19 works typically as follows. The RNA section, mRNA section, significant idea for generating a sequence encoding a critical protection of sRNA COVID-2 sequence used for templating and generating of the mRNA vaccines. Let's go for next slide. mRNA formulation. The mRNA encoding a spike protein that formulated in a lipid nanoparticles. This nanoparticle protect an RNA and produce a driver into a cells of a vaccine administrators. The vaccination. The RNA vaccines are administered into an individual through an intramuscular injections, typically into an upper arm. One injection, lipid nanoparticle delivered into mRNA into cells and in an injection site. Cellular uptake. The cellular take up a lipid nanoparticle containing mRNA once inside a cell. The mRNA serves a template into protein synthesis. The protein production. The cell mixturize a read of the mRNAs and produce a copies of a cell protein encoded with a vaccine are displayed into the surface of the cell. The immune response. The immune response system is recognized as spike proteins as a foreign and amounted into immune response. This includes a production of antibodies and specifies a target of spike proteins, as well as action of other immune systems and cell surface of the T cells. The immune memory. After the vaccination, the immune system is treated as a memory of spike proteins. This vaccination is induced as the latest sRNAs of an immune quickly response of a MOUNT3 response against the preparing of a reduction of each services of a COVID-19. Let's go for next slide. RNA vaccine for COVID-19 have a domestic highlights of as clinical trials have been authorized for the emergency use of many countries around the world. They offer a several advantages including the ability of rapid designing and manufacturing of vaccine scalability to production of an absence of the live virus and viral vectors which have an safety. However, challenges remain in the term of a disruption and storage of addressing of the pathogens. The DNA profiling of a DNA typically has a technical use identifying an individual based on the unique DNA characteristics. It involved in amazing significant results of a DNA of creating a genetic profile that as compared to another DNA samples for identifying the process. The DNA fingering are also highly accurate as a this, and also it have a DNA profile and profit of identifying a twins who are shared with a DNA profile. It has a revolution of a foreign synthesis science and have been instrumental in solving a content-less criminal cases as well as releasing of a worthless contradicting individuals. The additional DNA fingers are also used for various non-foreigns and also applications. The assets genetically testing are also aspects of a biology. Let's go for next slide. The DNA vaccine for rabbits. The rabbits are the viral diseases that affect wild animals and domestic animals, and it is translated into humans through an animal contact. It is classified into widespread of zoonotic diseases. The DNA vaccines are the rabbit present in the promising approach of RNA presentations, offering the advantages such as stability, easy production, and safety. The mechanisms of DNA vaccines for rabbits are the DNA enclosed with rabbit antigens. A DNA vaccines contains a smaller and circular piece of a DNA that encoded with specific antigens for the rabbit virus. These antigens are typically used for rabbit virus through RBC are circulated for the immune response. Let's go for next slide. The inner muscular injections. The vaccine is administered to viva injection onto a muscle tissues. Once inside a muscular tissues, the DNA is taken up as a beginning of a process of antigens. The antigen productions. Without the host cell, the DNA traveled into mRNAs which is translated into a rabbit virus as antiproteins. This antigen proteins are present on the surface of the host cell. The immune response activations. The presence of a rabbit virus antigens triggers a host immune system. This leads for the activation of a both cellular and human response, which is essential for fingering a rabbit virus infections. The productions of an RNA for a rabbit virus. The anti-selection, researching identification of selecting of antigens of rabbit virus that is almost effective as including as an immune response. The RNA proteins of a common choose due to the role of an viral attachment of an entire host cell. This DNA sections as encoded with the selection of rabbit virus and enclosed with a clone with a common vector service of delivering the vehicles for the variant of DNA vaccines. Let's go for next slide. The purification of recognizing a plasma DNA is preferred for a various technique used for immune response of high quality vaccines or production. The formulation of RNA diseases. The purifying of a DNA vaccines is formulated into sustainable delivery systems such as saline solutions or lipid nanoparticles to facilitating administration take-up of a host cells. The immunization and applications of the rabbit virus. The prevention of vaccination. The DNA vaccine is for rabbits or the administration of an animals is a risk of a rabbit exposures. The post-exposure of PCE. The vaccine of an is almost under the art of the use of the post-exposure of an PEC as induced of a bit or scattered of the animals suspend to carrying of a rabbit virus. This is complementary and tradition of the rabbit virus and rabbits are also administrations for RIG. Let's go for next slide. The advantages of RNA in a rabbit virus. The stability. The DNA vaccines are stable for a room temperature, essential and need for the cold chain store of transitions. The easy production. The DNA vaccines are also production for a recombinating of DNA technology, offering a scalable and cost efficiency manufacturing process. The safety. DNA vaccines do not contain a live virus, reducing the risk of the vaccine associated with and very administrative events. The challenges faced in this is while DNA vaccines are induced to store immune response, with a particular and large scale of the human population remains a challenge. Regulating the approach of a DNA vaccines required an extend data and safety for a long-term change and effect also. They may possess often very well-spread uses also. Let's go for next slide. The technique used in DNA diagnosis. The polymerase chain reaction (PCR) amplified the significance of DNA for analyzation. Next generation sequence (NGC) enable for compressing a genetic profile for a sequence of a quick and genetics. CRISPR technique is used for exploring the prevention and editing of a diagnosis for a genetic conditions. Let's go for next slide. The infection decisions of a diagnosis. The DNA diagnosis are key to identifying a pathogen causes of infection diseases. The techniques of PCR is detected with the various bacteria of and rapid editing of a timely treatments. This approach has been critically during of outbreaks such as COVID-19 pandemics. Let's go for next slide. The rare genetic disorders. The DNA testing facilities of a diagnosis are rare genetic disorders that offer for overlooked. Early and accurate diagnosis are significant for improving the management of these conditions. The genetic counseling can provide support and information of a facilitate families are the mere and rare of a genetic disorders placed in the crucial role. Let's go for next slide. The challenges in DNA diagnosis. The interpretation of a genetic data as a complex requirement of specifying equipment. The ethical consequences of a privacy and also a potential dismissal of a genetic information. The accessibility and affirmation of a genetic test remaining a significant barrier of many patients. Let's go for next slide. The future directions of DNA diagnosis. The advanced artificial intelligence are expected to enhance and interpret genetic data. The end of a myopic approach, we can know the more comprehensive understanding of the diseases. The content research necessary for explaining the applications of DNA diagnosis in various medical fields. Let's go for next slide. The applications of a cancer diagnosis. The DNA diagnosis is critical for identifying and mutual associated with various cancers. The lipid biopsy allows for a non-invasive identifying of eternal DNA for blood samples. Targeting of a theory development based on the significant and genetic methods of a turnover. Let's go for next slide. The DNA and diagnosis technique. DNA based on diagnosis allows for deleting pathogens of a genetic level. The techniques like a PCR, polymerase chain reaction, amplified for a DNA sequence for identification. These methods are usually critical for diseases of many outbreaks. Let's go for next slide. The case study of DNA diagnosis. The case study demonstrates the successful outcome of DNA diagnosis for personalized medical and meditations. The examples including for a periphetic are based on BRA mutations. This real-world application was highlights proteins and also for the improving a patient care through a genetic insights. Let's go for next slide. The advantages of a DNA vaccines. The DNA vaccines are stable, not require a cold state storage like a traditional vaccines. It can be rapid design for manufacturing in the response of any pathogens. These are safety profile for a favorable and also not involved in live pathogens. Let's go for next slide. The current applications of the vaccines. Several DNA vaccines have been developed into meditations and also medical for a livestock pets. That for first DNA vaccines approved for the human was the preventing of West Nile virus. The outgoing results are explored for DNA vaccines for diseases like Zika and HIV and COVID-19. Let's go for next slide. The conclusion part of this video. The DNA technology is and responsible of both vaccine development and diagnosis capability. Ongoing research can involve in a critical outcoming of existing challenges. This future of a DNA in the medical holding of significant promising of involving of public health outcomes. Thank you. Please like and share the video.